Well, good morning, biology of fishes. The morning being the morning of 18 October, I think, 2012. I think we have handled bioenergetics and its successors in a fairly adequate way. So I want to move on now with basically what's part two, which is now that we sort of have an understanding of some of the steady state components of the fish environment system, I want to talk about transient state in a little more detail, and with some reference to specific things that have to do with the fact that fish, like you and me, have to cope with a world that's not steady state, a world that's changing all the time, through time and through space. And that leads us to a consideration, first of all, to physiological management of internal environment. And then a little later, I'm going to complement that idea with the management behaviorally of environment. And we're going to do a lab. In fact, our lab 5 will be focused on behavioral regulation of environment, or enviroregulation, I like to call it. First, though, let's talk physiology. And so I guess I'm making the point again in A there that I just made, and that is that this is one of two major subsets of, and they're not completely separate, they overlap, of strategies for accommodating the fact that not all of the available space that's in the world ocean is appropriate habitat, and to some extent animals can extend the utility of the habitat by regulating their internal environments in the face of adverse or extreme external environment. And you and I do that too. You know, there's a whole segment of a course that I taught in physiological ecology that dealt with physiological thermoregulation and homeotherms. Fish don't do so much of that, so we will focus instead on the thing they do manage, which is mostly water and salt balance. That's probably the best understood part of adaptation biology as far as managing physiological environment. The idea in both cases, of course, is that you have to be able to somehow manage an internal environment that's different from the external world around you. You got to generate and hold the levels of environmental factors inside in a more normal or a more optimal range than the outside levels. And I'm thinking here, I guess, about as the factor E, which stands here for environment, I'm thinking about temperature, because not all components of environment have extremes both low and high. I mean, I guess one would argue that, for example, things like dissolved oxygen could be too low, but actually maybe they could be too high. And things like toxic materials certainly can be too high, but maybe they can also be too low, because a lot of things that are toxic at high concentrations are actually required at trace levels, as you know. Temperature, though, is a good example of something that obviously has distinct low and high values, and in between there is some optimum level that the animal tries to maintain, or would do best if it could maintain. Tries is sort of teleological, I guess. So that, the system functions best at some intermediate optimal level, and function declines as the level of that factor goes more and more extreme in both directions, and finally goes to zero, which I guess would correspond, if not in the immediate sense, at least in the ultimate sense, with a lethal level of the factor. Later on, we'll learn that the position of this curve is subject to change with acclimation in the individual. So, here's another kind of way of looking at it. On the y-axis is graphed the internal level of some environmental factor, and on the x-axis is the external level. And the idea is if the animal can regulate the internal level, then the level of the external environment that can be tolerated or over which performance is at least minimally acceptable can be extended. So if you take a look here, we're saying that from some limiting lower level here to some limiting higher level, that the animal really needs to be between EL and EH. But if it can manage, then it can extend the environmental level all the way down to EL minus, and its higher level all the way up to EH plus, and still keep its internal regime or milieu or whatever you want to call it in the optimum or in the survivable range. At this point, it's appropriate that internal and external levels be the same. I'm thinking about the fact that maybe so, but there can be an offset, as we'll find out in the case of salinity. So obviously, if you're a perfect regulator, then you bring this curve up so that it's essentially a straight line all the way across. And in concept, at least, the range of environmental values that can be tolerated or over which the animal can perform becomes infinite. Well, obviously that doesn't happen. But in the case of body temperature in mammals, it happens over a broad range. You and I can keep our core temperatures within a degree or so of 37 degrees over a pretty good interval of temperature, maybe not as much as you would guess. We have to do some behavioral things if we want to really extend that range, like putting on some clothing on the low end particularly. And then at some extreme level, of environmental value, the internal level fails, and breaks away to become the same as the environmental. So you can imagine a flat line going across here, and then at some point it goes straight up on the right side, and over here it goes straight down, and the animal comes to equilibrium with the level of external environment. But anyhow, if it were flat, we would say that the animal is a very good regulator of that particular environmental variable. And if the line is 1-to-1, then we would say it's a conformer. So ordinary fishes, particularly smaller ordinary fishes, are good thermal conformers. They are poikilotherms, as we learned in lab. So their core temperature values are pretty much 1-to-1 with the external temperature at steady state. In the case of fishes, if we want to see a good case of regulation of internal environment, then probably the first place to focus is on salt and water balance, because most fishes are good osmoregulators and ion regulators. If you want to read about this in the text that I point you to, it's chapter 24, Bond's book. But I like the little book by Linwood Smith from University of Washington called Introduction to Fish Physiology back in the 80s. I like his treatment of this topic. Let's talk about some background that's probably already familiar to most of you. First of all, how do you measure things that are related to concentration of osmotically active materials? The two measures that are commonly used are osmolality and osmolarity. And the way I make the distinction is that we have osmolality. We have a 1 osmolal solution if we have a solution that's 1 mole of perfectly dissociated substance to which is added a liter of pure water, or 1 mole of substance in 1 liter of water added to 1 liter. Get the liter first and then put the substance in, mix it up. And that's a 1 osmolal solution. But if you take a mole of substance and add water until you get a 1 liter volume of mix, then you got a 1 osmolar solution. These properties of solutions that depend on the numbers and not necessarily the kinds of particles, associating particles, are called colligative properties. And that was the osmotic concentration up there measured one way or the other. Here's some other things that are related in a 1-to-1 way. One is freezing point depression. The higher the osmotic concentration of a fluid, the lower its freezing point, and that's expressed usually as delta F. It's important to keep in mind that the F stands for freezing and not for Fahrenheit. You can measure freezing point depression either centigrade or Fahrenheit, of course. Usually it's centigrade degrees. The more osmotically concentrated a solution is, the higher its boiling point. So, water boils, pure water boils at 100 degrees centigrade at 1 atmosphere. But if you add solutes, the boiling point goes up. The vapor pressure is depressed. That's the pressure that's exerted. If you imagine a solution in under glass or contained in glass, at steady state, the pressure that the particular substance exerts in the vapor phase would be lower if there's more solute in the water. And then osmotic pressure, and that's an actual pressure that can be a hydrostatic pressure that reflects the fact that water tends to, like anything else, diffuse with its concentration activity gradient, meaning that it diffuses across permeable membranes to places where water is less concentrated. I think we make a mistake by trying to talk always about the concentration of the solute instead of the concentration of the water. Obviously, if there's more solute, there's less water. And if you have a lot of osmotic material, osmotically active material, then water diffuses in and can exert a fierce, 100 atmospheres possibly in the case of guard cells on plant stomates. The stomate guard cells, they develop turgor because of osmotic pressure and close and open. And it can be a very high pressure. You probably all I don't know if they do those experiments anymore, probably safety considerations prohibit it, but I remember in grade school, exposing blood cells to distilled water and salty water and watching the crenulation or the shrinking, the shriveling, I guess is the right word, of the cells in the case of salty solutions, and explosion caused by osmotic pressure exceeding the limits of the membrane in the case of a dilute, putting the cells in distilled water. There's also another property that I guess you could think of as colligative, and it's the one we usually label salinity. And as long as the salts are in fairly low concentration and dissociate, then you get a pretty good one-to-one with osmotic pressure. And there's a little table that you can't find just the first time you look any place. At least I never could. So I finally put it together, and I commend it to you as a non-official source, but it's a nice way to relate the salinity as we usually express it in parts per thousand of total dissolved solids with the freezing point depression and with osmolality measured in osmoles or milliosmoles per kilogram. And so over the range of salinities that would be of most interest to most aquatic biologists, 0 to 40, freezing point depression goes from almost nil at 0, obviously, to 2.35 degrees below 0 at 40 parts per thousand. And notice that at 35 parts per thousand, which I've told you is pretty much the general average salinity of the world ocean, maybe it would be a little lower than that if you consider the entire ocean, including the Arctic and subarctics. But at around 2 degrees, saltwater or seawater freezes at about -2, and that's something I think we've already talked about. And then over here is the osmolality, and notice that it's 32 parts per thousand is 1,000 milliosmoles, or 1 osmole. And I think that's why people like to say 32 is representative of seawater. Makes an easy translation to osmolality. By the way, this relationship is restricted to normal seawater, the mix of salts that occurs in normal seawater. If you start looking at saline groundwater, for example, from the Permian Basin, then the relationships don't hold. It's still pretty much 1-to-1, but the numbers are different because the composition of that water is different from seawater. So, what I've done over the years is try to refine and condense this information to the point where it is, I think, fairly accurate, but broad and simple. And so I want to talk about it as a problem of water ion regulation, and then talk about the solutions that fishes have brought to the problem. The internal salinity of freshwater and saltwater teleos are not that different, but freshwater teleos tend to be a little lower, around 8 to 10 parts per thousand, as measured or expressed as salinity, and that's the way I'm going to express it. Marine teleos a little higher, 12 to 15, but it's easy to find cases where the two overlap. Particularly if we're talking about euryhaline forms. But I think if you were talking about primary division marine fishes that never get within sight of fresh water, things like the reef fish community, the 12 to 15 out here would be probably not too bad, and then this would be strictly freshwater forms, like the perches, for example. I used to say centrarchids, but centrarchids can dabble in the salt a little bit and occur in coastal waters, so I'm not sure they really are as strictly freshwater as perches, the Persidae. So those are the levels of salinity that evolution has designed these forms to operate at most efficiently and optimally. And that makes for a problem because the freshwater habitat is less salty and more watery than the blood of freshwater fishes. And the marine habitat is a lot more salty and less watery than the blood of marine teleos. So freshwater teleos have the problem of having blood fluids that are more concentrated in salts, diffusible, or more. What am I trying to say? Not diffusible necessarily, but more dissociating than the water. So they're hyperosmotic. The freshwater teleos have blood plasma that's hyperosmotic relative to the water they live in, or the water is hypoosmotic relative to their blood. And so the problem they have is trying to scrub rare ions from the water and then keep them against the diffusion gradient for ions, and at the same time resist the diffusion in the opposite direction of water. Most folks use the word osmosis to describe diffusion of water through biological membranes. So I guess osmosis is a special form of diffusion having to do with water particularly, but ions also diffuse. Saltwater teleos, on the other hand, have just the opposite set of problems. You know, they got blood that's hypoosmotic relative to the ocean they live in, and so they're constantly losing their water to a saltier world and at risk of gaining too many ions from that saltier world. So what do they do about it? Well, in the case of freshwater teleos, the gain of water by osmosis is resisted by their having a skin that's relatively impermeable to water. And they share that actually with marine teleos too. Their skins are also impermeable. And even if they're like catfish and don't have scales, that skin is still impermeable. If you have scales too, then that adds to resistance to flux through the skin. It's said that freshwater teleos resist taking water through the gut by not intentionally drinking water. But there's no way to avoid taking some water into the gut when they are eating food. But most of the water enters freshwater fish where the exposure is maximal, and that's in the gills. And so the water that leaks in by diffusion or by osmosis at the gills is pumped out by is pumped out by a well-developed kidney that can produce a large amount of urine that's relatively dilute. The words, I guess, that are always used in the textbooks are copious amounts of urine. 5 to 15% of body weight per day sounds pretty copious to me. And the urine is dilute, but it's not dilute compared to the water in which the fish are excreting into. It's got a freezing point of depression of minus 0.05 degrees, whereas the pure water, if it's pure, is 0 degrees. So that urine is still hyperosmotic to the water. And so there is a lot of salt lost in the urine. Not nearly as much as it would be if the fish were just excreting, if it just took its glomerular filtrate from its kidneys and passed that straight out, without some recovery of ions. And of course that's what goes on. If you looked at kidneys, you know about the tubules and all of that, and the loops of Henle and things. I don't talk about the anatomy, just about the overall picture here. And the overall picture is that the freshwater, the teleos kidney is well developed, lots of nephric units, and very efficient at recovering ions from the filtrate from the blood, which is basically the plasma that goes through the glomerular, through the what? I'm trying to think of a word. Help me here, Allison. You know all the words. The bulb, Yeah, you do. You know, but as soon as I say the word, you'll say, oh, yeah, that's what he meant. Bowman's capsule is what I'm trying to say. Oh, see, I knew it. Actually, I knew it. I was just, no, I wasn't. I couldn't remember the word. Bowman's capsule, you know, you've all seen the picture, you know, with the little hand, you know, kind of structure, and you got this glomerulus of this network of capillaries in it, in the kidney, and it's leaking out all this plasma, you know, with the pressure, the blood pressure, and that's going through into Bowman's capsule and becoming the filtrate, the glomerular filtrate, and then from that is reabsorbed, scrubbed, the ions, and put back into the blood, so that what you're left with is just mostly, you know, water, but it's pretty salty water. It's got a freezing point that's on up there, and it's important to realize that a lot of salt is lost at the kidney with the water. That loss in the urine, which is very substantial, as well as the loss at the gills through just diffusion of the ions into the water from the blood at the gill, has to be countered by active processes. Mainly it's active uptake through cells that historically have been labeled chloride cells, although they actively absorb all kinds of mostly monovalent ions, both positively and negatively charged. So there's these chloride cells in the epithelium of the gill that are specialized to absorb ions, and then also freshwater fishes take advantage of the work that other animals have already done, and I think that's kind of a neglected aspect of the whole process. You know, that a lot of, you know, if you're a fish and you're eating another fish and the other fish has 8 parts per thousand body fluids, then you got the 8 parts per thousand along with the nutrients. And I guess I can have more to say about that in a bit. So we got absorption in the food and uptake at the gills through the chloride cells, and that's the way freshwater teleos, that's pretty much the summary statement of how they deal with the fact that they live in a watery world that's where salt is scarce. Marine teleos, you know, have just the opposite set of problems. They're always at risk of drying out. You know, sometimes the people talk about the marine desert from the standpoint of water. You know, water everywhere, not a drop to drink. Actually, marine fish do drink it, and that's probably the one thing about the whole story here that's kind of counterintuitive. But I'll get to that. So what marine teleos do is minimize the loss of water and the gain of ions through having the same kind of impermeable skin and scales, or at least similar in function, to freshwater fishes. And they certainly don't want to throw away any water because they're having a hard enough time keeping what they got. So their kidneys are not designed to produce much urine. In fact, they produce relatively little urine, and some truly marine forms, the primary division marine fishes, have kidneys that are said to be aglomerular, you know, really without much function at all. And all the marine fishes tend to have kidneys that are reduced in size, and the nephric density is low, and the individual nephron units are small. So marine fish don't produce a lot of urine. They lose their water anyhow, though, mainly through their gills, and they replace it by a combination of drinking seawater and by actively excreting ions at the gills through chloride cells. And it's argued whether the chloride cells that do the excreting in marine fishes are different from, or just change their polarities from those in freshwater fish gills, whether they just recruited a different subset. Because some marine fishes or some estuarine fishes move regularly from fresh into salt water over periods of just a few hours, and the functions of these chloride cells just switch. Things like mullet, for example. Back to the drinking of seawater, though. The idea here is that marine fish can drink up to 35% of their body weight a day in seawater, and then absorb 75% of the water through the gut mucosa. And this takes energy because they're doing it against a gradient in a holistic sense. So what they do, the trick is that they actively absorb monovalent ions in the interior part of the gut mucosa, raise the resulting osmotic concentration of the mucosal blood to a level higher than the seawater that is there after they've removed a lot of ions from it, and then passive diffusion allows water to be absorbed into that mucosal blood. Well, so you've had to do some work to do that, to absorb those ions in the gut in the first place. And then you got to get rid of them at the other end at the gills, so you have to double dip. The divalent ions aren't so much subject to that active uptake process. And so things like calcium and magnesium and sulfate tend to be passed on out with the feces. And in some situations, the fecal slurry that's produced by fish in really salty situations, is almost pasty with these salts. It's pretty highly concentrated because you've only got 25% of the water left with all of the divalent ions in it, or most of them. So I had a thought that I was going to mention, but I didn't. I've lost it now. Let's see, what was it? Maybe Valdemar will remind me of it, because I saw him start to put his hand up. What was your thought, Valdemar?
I have one question for you. On an isosmotic condition, how much energy do we save on osmoregulation?
Under isoosmotic conditions, how much energy would be saved? And the answer to that question is, I think nobody knows, and I'll talk about that in my last slide in this session, is the energetics of osmoregulation. All I know is that biophysics analysis does not give the right numbers. I'll have something to say about that. Let me defer on that question a little bit. I'm not going to answer it for you. I'm going to tell you that nobody can account for the amount of energy that it takes. I don't think at this point. So I guess the main thing I want to emphasize here is that marine fish are doing something that sounds counterintuitive, which is they're drinking water and they're putting that in their gut that it is saltier than the blood measured over the whole body. And so how in the world, there are no, I guess the point I wanted to make is there are no bulk transport pumps that pump water, I mean, the only pumps that do pump water are bulk, I should say. There are no molecular level active devices for moving water, as far as I know. The only way to move water is with bulk pumps like hearts and gills. But you can't actively excrete water or actively absorb water. What you do, if you're a marine teleost, is you actively absorb ions into the gut mucosa, lowering the concentration of those salts in the water that remains, raising the concentration locally in the blood of the mucosa, until finally the gradient favors diffusion of the remaining water that's still there in the gut into the blood. Then the blood flows to the gills, where those same ions that were actively absorbed you have to turn around then and use your chloride cells to actively excrete them back into the sea. So it's working twice in order to do the job. And maybe it was here that I wanted to say that this idea that fish, marine fish that have evolved to have physiologies that function best at way above fresh water, like red drum, may benefit from eating prey that have salt already contained in their blood fluids. And this is not a new idea or my idea. It's an idea that I think has been around a while. But some observations that we were involved in sort of brought it home to me, and that was that marine fish like red drum that have been transplanted and stocked into freshwater systems like these power plant cooling lakes in Texas, Brawny down in near San Antonio, and Fairfield Reservoir, Big Brown Power Plant up near Fairfield, Corsicana. In some of those situations, people have marveled at the very good growth rates of red drum. So, getting kind of to your question, Valdemar, what kind of energy cost is associated with osmoregulation? You'd have to wonder if it works you hard to regulate your salt balance against a stronger gradient than you normally have to deal with. How would you grow fast? And that was the question, and I think the answer that has come up independently in a number of different places. And when I say grow fast, I mean relative to coastal situations where the same species is. The idea that's come along is that, hey, these fish are actually maybe eating for salt. And a side benefit of all that consumption is that you got a lot more nutrients than you might otherwise have. And maybe there's an advantage, too, of living in situations where one of the dominant prey items is also a cold-intolerant form, tilapia. Most of these power plant lakes have been stocked with tilapia years ago, and they get kind of slow when they get cold in this time of year as the water finally cools off, and a lot of them die over winter and are forced into the outfall areas from the power plants as thermal refuges. Well, so you can imagine a system where both the predator and prey are forced together in a high-density situation in a heated effluent outfall. And so, these red drum are finding pretty plentiful prey. Maybe that's how they can achieve such good growth. I think I made the point before that Fred Fry told us that lethal factors are not the same as limiting factors that have gone to extreme values. And maybe the proof of that is these red drum in power plant cooling lakes. Even though they are growing at rapid rates, they're very fragile and almost totally intolerant of any handling. So fishermen are I think, of the opinion that if you're going to catch a red drum in one of these power plant lakes, you might as well fillet it and take it home, because it's not going to live if you put it back in the water. I mean, anytime you break the barrier, the mucus, it's stressed, and there's probably osmotic stress that's aggravated by other kinds of stress. The fish develop fungal infections, and they don't live usually past that experience. So, there's another installment to this. Oh, I haven't finished telling you all the story. The rest of the story is we said to ourselves, well, if a fish like a red drum is eaten for salt in freshwater systems, might one add some salt intentionally to prepared feeds when you have these fish in freshwater systems, and would that benefit them? And so we did an experiment. I think Dr. Steve Craig was the guy that did the experiment, and with Dr. Gatlin and me. And we fed red drum ordinary pelleted feeds that had an additional salt amount added in both freshwater and saltwater situations, and the result was just the one that you would predict if the hypothesis were true. The fish that were in the low salinity environments grew better, performed better when they were fed the salt-enhanced feed. And more recently I've been thinking about the opposite situation, and I thought it was an original thought. But, there are no original thoughts. There are only thoughts that somebody else had, and you heard them somewhere, and you sort of subconsciously processed them and accepted them as your own. And I want to give credit to one of your predecessors. Some of you will know Randy Zedzik, maybe. He was in this course in the spring 2011. And I had forgotten all about this. I had been talking to Dr. Gatlin and Dr. Vega about research that might be done to deal with increasing salinities that we're seeing in the Texas bays, with the dry times that we're in, the salinities that Robert and his colleagues, Rodney Gomez, who runs the hatchery at the Marine Development Center, are facing are up in the 60s, 70s in those ponds, and they're having a hard time growing fish at those high salinities. And so we were thinking about all kinds of strategies for dealing with high salinity. One of the strategies you'd like to use, but you can't because it's not cost effective, is just pump a lot of fresh water and dilute those ponds. Just the cost of that water would be outrageous. But one way to maybe have some of the same effect would be to feed fish an artificial ration that's intentionally hydrated. Feed them a wet feed, and see if they will get water from that feed at lower cost than living and trying to get it from the pelleted feed that they're being fed now. I mean, they're eating some forage, but when the fish get close to harvest, the ponds usually don't produce enough natural forage, so you have to add supplemental feeds. Well, anyhow, it was an idea that I was promoting, and last night, folks, I was playing back last year's recording of this presentation, which was better than this year's, by the way. A lot of times they are. I'm disappointed that I'm not improving, But toward the end of that session, I responded to a question from the group that was just like you sitting here in the room with me. And the question, fortunately, I identified the speaker. I said, "Yeah, Randy, you have a question?" And the Randy I checked is Randy Zedzik, one of our undergraduate majors. And he said, "Yeah, Dr. Neal, I was just wondering if it might work the other way around. You've been talking about these fish in these power plant cooling lakes and freshwater, and you've been talking about the possible benefits of feeding fish salt-enhanced feed. Might it be possible to feed fish in too salty situations, salt, free feed?" Well, I don't know about that. I'm not sure. Palatability issues get to be involved at some point, I'm not sure that reducing the salt in pelleted feed to zero would really give you much of a gain, but adding some water might. Now, wet feed is a lot more problem. Anybody who's ever messed with fish feeds knows that it's really a lot easier to extrude and dry pelleted feed than it is to make a wet feed like Oregon Moist. That was a commercial feed for a while that was used for salmon. And it was a wet, spongy feed. So I don't know, I'm not sure what the technology would be, but we're going to pursue it. I think Dr. Gatlin's excited about the notion of trying to explore this idea. And I want to give Randy the credit on the record for coming up with it. It's a logical idea. I don't know if it'll work, but, that's the way research happens. People have ideas, and somebody does a controlled experiment to test the hypothesis that emerges, and if it works, progress is made, and we know more than we knew before. I guess we know more in either case. If it works out like you kind of hope it would work out from an aquaculture point of view or fisher's point of view, then maybe you've really made some progress. Aren't y'all excited about that? That's pretty neat, huh? The idea that started in this classroom. So Allison, I can see the wheels turning. Allison's our salinity expert here, at least one of them. So what about these anadromous and euryhaline teleos? And I'm not going to talk about the other side of what diadromous fishes, or fishes that spend some of their life history in one kind of regime and then the other part in another regime. Freshwater, saltwater, marine freshwater. Diadromous fishes come in two groups. You know, there's the catadromous forms that do their spawning and early development in freshwater and then do their growing up in the marine world and spawn there or reproduce. They grow up there and then come back to freshwater, either close to freshwater or actually into freshwater to spawn. And I'm thinking of red drum. And then somehow or the other, the larvae find their way into the fresh. In the case of salmon, of course, they do the whole work. I mean, the parents do all the work. But in the case of red drum, the spawning seems to be occurring at full strength seawater in the passes. But somehow or other, a combination of active processes and passive drift and modulated drift maybe moves the larvae, the developing larvae, into low salinity water, if there is any low salinity water around for them to move into. So those are what I would call anadromous forms. Then you got catadromous fishes like the eels, which, you know, they migrate to someplace like the Sargassum and spawn there, and then the young eels get on the merry-go-round that is the Gulf Stream and eventually come into freshwater rivers and do their growing up, either way upstream in some cases. I remember catching American eels in streams in the northern Ozarks in Arkansas when I was an undergraduate. That's the females. The males, I don't think, make that whole trip. You know, they're lazy. They hang around on the coast. They don't go all the way upstream with the females. I don't think they meet on the way back, apparently. You know, the whole eel life history thing is still, I guess, something of a mystery, exactly when and where and how. I'm not sure anybody yet has actually caught eel spawning. Maybe not. Maybe they have. And then there's a South American salmoniform group, Southern Hemisphere salmoniform group, not necessarily all South American, called the Galaxiids, that have a catadromous pattern, you know, kind of opposite their North American, North Pacific, Northern Hemisphere group. So anyhow, anadromous, diadromous, I guess, and euryhaline teleos. When I say euryhaline teleos, I mean ones that are living in the coastal zone at a variety of salinities ranging from nearly fresh to nearly full strength, or maybe even more than full strength seawater. Like all of the cyanids, I would consider, at least the marine ones, to be euryhaline. And what they do is they operate, in the appropriate mode. When they're in fresh water, they excrete water like a freshwater fish, and they through their kidneys, and they hoard ions. And when they're in the ocean, they do the opposite. They excrete ions and hoard water. The physiological adjustments can happen over a matter of just a few hours or maybe a few days. The chloride cells do the flip-flop, or different sets are recruited, and the kidney changes its function, reduces urine production as the fish moves into the marine habitat. And this drinking either starts or stops. I had a former student, Dr. Jim Bryan, who did some modeling of red drum osmoregulation, published a paper on that in case anybody would like to see it. And one of the conclusions he reached was in order to account for the observed blood ion concentrations and osmotic values in the blood, the specific ion concentrations and metabolism, you had to invoke an argument that there was a lot of overlap in functions between the marine and freshwater phases in these fish, that they're producing urine when they shouldn't be. They've already moved out into marine systems and they're still producing a lot of urine, and they're drinking when they shouldn't when they're in freshwater. They overlap the drinking and the urine production functions. So I don't know if anybody's explored those ideas. Yes, Alejandro.
This might be a silly question, but going back to the energy equation, when we look at the AS, I remember that it varied to, fish size, weight, and temperature. So you could also conclude that there's no variation with environment in that case.
Well, let me just get to the end here, and we'll see my guess about what the situation is. And I'm not sure it's right, but if you look in your handout that I gave you, the next to the last slide will show you some metabolic rate as a function of salinity for different groups. And I want to spend a little time on that. But I'm the first to admit I really don't know what's going on with this. And I do know that the biophysics modeling is really not giving... You can't account for all the work that's being done, apparently. There must be some couple things happening in addition to just the pure maintenance of ionic differentials. The endocrine system has to be important. Prolactin, the freshwater hormone, some people have called it, and cortisol produced by the adrenal, and even thyroid hormone, I guess, get involved in this switching process and regulating things like chloride cell function and kidney function. So elasmobranchs do a whole different set of things. If you needed any evidence, you probably have plenty by now that elasmobranchs and teleos are way far apart, in their origins, because they have very different responses to common problems. There've been a lot of, and one of those examples is osmoregulation and ion balance. Marine elasmobranchs, as I've already told you, have a blood osmotic concentration that's very similar to seawater, maybe slightly hyperosmotic to seawater even, as opposed to being way hypoosmotic in the case of the marine teleos. And the way they achieve that is not by raising concentrations of things like sodium and chloride, but by retaining urea and other nitrogen metabolites like this trimethylamine oxide. That's what TMAO stands for. The marine forms actively excrete salt ions with a rectal gland and a kidney, and they have gill chloride cells as well. Freshwater forms pretty much operate just like freshwater teleos. They don't keep the urea in their blood. They go ahead and get rid of it. And it looks like all of the processes in the freshwater ones are basically mirror freshwater teleos. So the one thing that's really different is this urea retention. Cyclostomes, again, it's pretty obvious, I think, to everybody that studies these fishes that lampreys and hagfishes are totally different critters, even though they both are grouped together as the round-mouthed fishes. Lampreys are perfectly good ion osmoregulators, and they operate pretty much like teleos. And lampreys, of course, move from freshwater to marine and back again. And when they're in marine habitats, they operate like marine teleos, and when they're in freshwater, they operate like freshwater teleos. Hagfishes, on the other hand, are all marine, and seem to be pretty much osmoconformers, with the exception of some of the negatively charged ions that they seem to regulate. So they operate more like some invertebrates do, and very primitive invertebrates at that. Most of the invertebrates are pretty good osmoregulators. They have water-permeable skin and blood that's essentially isoosmotic to the seawater medium. Hardness ions figure into osmoregulation in an important way. Calcium and magnesium, which are the dominant hardness ions, not iron, by the way, folks. It's not iron, it's calcium and magnesium. Calcium and magnesium reduce the permeability of biological membranes to water. And so hard water tends to make it easier to manage either gains or losses by diffusion of water through things like gills. Seawater is hard, so there it's not much of an issue under normal circumstances, but freshwater, of course, is highly variable between being very hard, like in the limestone country, the Texas hill country, for example, and soft water. So in soft water, there's not much hardness, and so there's more work involved in maintenance of salt and water balance, and this is something that every fish hauler knows. They tend to try to do things to increase the hardness of the water, add a little salt maybe to make the osmotic stress less. Hardness is especially important in adapting marine fish to quasi-freshwater habitats. So you better put some some calcium carbonate in fine form into your marine aquarium if you're gonna keep marine fish and lower the salinity. So here's a summary that I put together at some point that tries to bring all this together. At least if you had to study something for a final exam, you might want to look at this little chart. I got fish groups, teleos, freshwater, and freshwater sharks together, and then I got saltwater teleos and saltwater sharks. Environmental problems. Too much water and too little salt for freshwater, and too little water and too much salt in marine water, marine systems, marine fish. And then the solutions I've organized according to the location: the gills, the gut, the kidney, the blood, and the rectal gland. And the only thing that the rectal gland is really relevant to are marine sharks. Urea retention is also relevant to marine sharks. And then the kidney functions and the gill functions and the gut functions are pretty much as I laid them out, so you can read the description for yourself there. A little note down here: all the groups lessen diffusive flux via relatively impermeable skin and scales. Okay, here we come to the problem of work, metabolic load, optimum salinity. And for euryhaline forms, it looks like the optimum salinity tends to be about the same or slightly less than the isoosmotic salinity, which is typically around 10 parts per thousand. In other words, if you put them at all kinds of levels of salt water, experimentally, they perform better around 10, and that's true for cyanids, and it's true for probably for shrimp as well. For strictly freshwater marine forms, the optimum salinity tends to be about 0.3 parts per thousand, a little more than, you want a little salt in the water. You don't want it less than 0.1. From 0.3 to 1 part per thousand, and for 30 to 35 for marine forms. Those are the optimum levels of salinity. In terms of performance, but these values are well below, in the case of freshwater fish, and well above, in the case of marine fish, the isoosmotic level. And then I had a whole bunch of stuff on the Goldman equation and things, but I figured people have seen enough equations, so I've just reduced that to this statement. Metabolic work exceeds that you would estimate as the energy required to maintain the electrochemical differences that are observed. Way exceeds. I worked it out for a lot of different forms, and it's a factor of 50 or 100 times more. So I don't think anybody knows where the work is being done. It's obviously associated. It's not the strict maintenance of electrochemical differences. There's an equation called the Goldman equation that describes that work, but it doesn't account for the observed results. And the observed results are basically these. Here is my attempt to show and account for the optimum salinities for the different groups in terms of metabolic rates. And I've got active and standard rates shown in red and green, respectively. And then the blue is the difference between the two, and that's the metabolic scope. And so what this says is that freshwater fish have an optimum salinity that, they have a relationship between standard metabolic rate and salinity that probably reaches its minimum near the isoosmotic point. But the active rate looks like it peaks at very low salinities, around 1 or 0.3. And the consequence is that optimum salinity, in terms of where metabolic scope is maximized, tends to be, say, from around 1 to about 5. And then it falls off rapidly. And freshwater fishes don't do at all well above about 8 or 9 parts per thousand salt. In fact, that's the usual limit that you would find things like largemouth bass in an estuary. Interestingly enough, it's also the limit for cattle. Livestock folks, ranchers out in West Texas, know that you don't make your cows drink water that's saltier than about 8 or 9 parts per thousand. They'll drink it if they have nothing else, but they don't do well. So I don't know if there's any, I don't know if that's just coincidence or what, but I think the same value of salinity is pretty much the upper limit. And you're not going to get spawning in freshwater fishes at 8 or 9 parts per thousand either. That's going to occur more at where scope is maximum here at probably 1 to 3. Euryhaline fish, or marine fish rather, have their minimum standard metabolic rate probably in right around 25 or 30 parts per thousand. But it looks like for some reason that the maximum rate is pushed to the right and is more near like full-strength seawater, and the consequence of that is that they have their maximum metabolic scope nearly at full-strength seawater. And again, you don't get marine fish to spawn at low salinities. People have tried to get things like red drum to spawn at low salinity without success. They seem to be, 25 or somewhere 28 is probably the lower limit. So it looks like, I've got my true, I'm talking red drum now, and I should be talking marine fish in general. It looks like metabolic scope goes to 0 around 40 parts per thousand for truly marine forms, and probably about 18 parts per thousand on the other end. And then the euryhaline fishes, they just stretch the whole thing out and cover this middle ground pretty well. Probably the minimum standard metabolic rate is somewhere in the upper teens or around 20. But it looks like to us that the maximum active rate is probably below 10 in the case of euryhaline forms. The consequence being that you have maximum performance right around 10, but it doesn't deteriorate much all the way up to full-strength seawater. And once you're above that, then it falls off. And Allison will probably help us fix the end of this curve. It may even go further out to the right than I've shown it here. I'm thinking red drum when I did this. So that's the picture I see, and you got to wonder about, what's going on in urihaline forms. It looks like they're sort of mixing.
Past 60 it drops off.
And past 60, Allison said, is where you lose performance to zero. Well, my point I was making before you made that comment is it looks like they've got the best of both worlds. In fact, they're covering this middle ground where nobody lives. Neither freshwater nor marine fish would do well around 15, truly marine. So I'd appreciate anybody that knows the details of, and I'm speaking here to folks who are not just in the room, but people who are out of the room as well. If you know the details of performance of some fish at different salinities, I'd like to hear about it to maybe adjust and I may have to adjust this graph. Now you got to tell me, is it because active metabolic rate falls off at a higher salinity than I've shown, or is it because there is not this abrupt elevation in standard rate that would account for the fact that metabolic scope goes on the way out to upper 60s for these urihaline forms? What would you guess?
Well, it's actually a little bit of both. One, you get to the point where you can't proliferate any more chloride cells. So
you can't recruit any more chloride cells when you get to...
Right, those are mitochondrial rich, so you need more energy for them. Second, once you get past 60 parts per thousand, the oxygen load in the water starts to plummet.
And there's...
So you just can't pull enough oxygen out.
There's another good point. So 2 points that Allison made I want to repeat for the record because I'm not sure we picked you up well. One of them is that sooner or later you saturate your capacity to recruit chloride cells, and whereas you'd like to have more to do more excretion at these real high salinities, you can only pack so many into a gill. And second, the amount of oxygen in solution at these real high salinities goes down, the solubility being a lot less. And so, it's a combination of extra work to be done, but not enough metabolic resource to do it with. I like the sound of that. The sound makes good sense to me. What about these low salinities? There's plenty of oxygen there. There's more oxygen down there than there is at the optimum salinity.
I agree with your low salinity graph.
So what is it that causes standard metabolic rate to increase at low salinity?
Proliferation of chloride cells, because they're mitochondrial rich. So as you increase your salinity, you have to start creating these chloride cells.
As you increase or decrease?
As you increase your salinity. So
well,
you go from zero up, you slowly start building chloride cells, and those require a lot of energy. And then you have lots of mitochondria in them. So you have to start working and getting more oxygen in your body in order to create these chloride cells.
Well, I heard the argument, but it sounded like you were still arguing about increasing salinity. But what you're saying is as you go down in salinity, you have to also increase your chloride cells because you're going to need to scrub more rare iron from the water, so you have to work harder.
No, kind of. When you've got it right here on the 10 to 0, it decreases.
Yeah, and that's why.
Yeah.
That's why. It's because you're saturating your chloride cells on both ends.
Right.
On the one end, you're saturating them because you need to excrete more salt, but on the other end, the low end, down here, you're needing to absorb more salt, so you have to have more chloride cells, more work. Okay, I guess that would that also would that explain both standard and active? Do you think?
Yeah.
Okay. Does anybody disagree? Do you disagree with the general shapes of these curves?
No, I think it's pretty good. I think the graphs I sent you might be helpful. I think I sent you some data.
Yeah, I need to look at that. That was for pupfish or what?
Yeah, minnows and western mosquito fish.
Mosquito fish? Yeah. Okay. Thank you. I need to look again. All right, well, we're going to wrap it up. We're at the end of the class, and that's just what I had hoped that would work out this way because I didn't want to launch into new material today, even though I went ahead and posted the handouts. So we'll see you guys on Tuesday of next week. I hope everybody is getting the data that Donovan needs to send out to the whole group. If you're waiting on those data, let me point out to you that you don't need the data to do about 90% of the work. You know, you're going to be doing about 90% of the follow-up just with your own data anyhow. So don't let it keep you. Don't let waiting for the data hold you up, is my point. The other point to be made is that all of you will do well to get all those points from lab. All right, so see you next time. Take care.